Okay, so welcome to the 17th meme seminar. Today we have Sandra Lorena Ament-Velásquez. I believe she goes by Lori.
Yes.
And she is originally from Mexico, did her bachelor's in biology at the Faculty of Sciences at UNAM, same as Diana, I think. And then after meme, she worked for a bit at the University of Montpellier, started a Ph.D. in Zurich, and then switched over to Uppsala, where she recently finished her Ph.D. and then is now a postdoc researcher in the same lab, I believe. And she is working on fungus, and that's all I know. So go ahead.
Okay, thank you. I'm very curious as how you knew that I was in Zurich.
I think it was like it's on your research gate, maybe.
Oh my God,
one of those
I have
things.
to remove that. Yeah, that's a sad story that we can talk about later. But yes, in the end, I came back to Uppsala. So as I was explaining before, I had a little metaphor here in my cover slide, but don't worry too much about it. In fact, I think I have a lot of random pop culture references in the presentation. And if you don't understand, or if I'm going too fast, please do interrupt me. I prefer when people ask questions while I'm explaining things rather than wait till the end. So yeah, I thought that I could start, if this thing will let me, yes, just as everybody does, explaining my meme history. As you said, I did my bachelor's in Mexico. And then when I started meme, I went to Groningen. From there, I went to Munich. Then I did my first master project in Montpellier, and then my second in Uppsala. And during that time, well, when I was in Mexico, I was already working on fungi, on lichens. But during meme, I had the chance to also work on other organisms, as most of us do. And in my case, I was working on marine invertebrates, really fun cases also. But now for my PhD that I just finished, I was focusing on a particularly weird fungus called Podospora. So can you see my mouse, actually, if I hover like this? Awesome. So what you see here, I remember the first time I showed a picture to my supervisor, she was like, what is that? This is the fungus. This is the fruiting body. And it's a picture I took in the wild. So to me, it's very precious because there's almost no pictures available. And that's where the spores are, basically. So for this talk, I thought I could just give like a mini review of what's going on in the field, because most of these findings came up in the last 5 years. It's like all very, very new, very fast changing. And yeah, and at the moment we're working on a review actually. So I thought that maybe it was timely to start thinking about this in general. So I will very briefly introduce genomic conflict as a concept. I'll explain what meiotic drive is. Maybe I don't know if you people are familiar with this actually. And after that, I'll just basically tell you most of what we know about the main fungal meiotic drivers that we know that are called the spore killers. And in the end, I'll just summarize everything, and maybe if you want, we can just speculate more. All right. So yeah, I wanted to start with this toy, Lego toy, because I feel like it's a good metaphor of how a lot of people perceive biology, even biologists themselves. I feel like we often think, like all the pieces fit together perfectly and everything comes together in cooperation. But I hope after this, after this talk, you're feeling that that's maybe not the case, and maybe it's like a mess, kind of. And part of the reason for that, this mess, is the fact that selection can act at many levels, right? I think we often are trying to think on, natural selection happens at the individual level, blah, blah, blah. But you can also have selection at the cell level, at the gene level. I don't know about the population level, maybe that's open to discussion. But the fact that you can have selection at different levels means that there could be conflict, right? And so that leads to the situation of having selfish genetic elements where these genes, by definition, try to increase their own frequency at the expense of the host. They might be neutral, but they basically don't care, right? Now, it is believed, or I think at this point it's a big consensus, that there's this antagonistic selection between the selfish genetic elements and other components of the genome, the host, if you will, and that this antagonistic selection leads to evolutionary change, novelty. And you can see that in ecological terms. You can imagine it, like with host pathogen races, and of course, very timely with our COVID situation. But it's very similar in a sense. And I think an important aspect of this is that selfish genetic elements were almost there from the very beginning of life. So they are everywhere. They happen all the time. We cannot escape them almost. And when people talk about selfish genetic elements, I think maybe most of you remember from your classes, or maybe you're very well aware of this, but there's the transposable elements, right? That's the classic selfish genetic element. And I'm not gonna explain much about this, but as you probably know, they are these pieces of DNA that can replicate by moving position in the genome. There's many classifications, but the typical broad categories is like the copy-paste transposons, like they just literally, they look like copy and paste. And then the cut and paste ones that cite themselves and then insert somewhere else. These transposable elements, or not all of them, but a lot of them, produce a specific signature when they insert called the target site duplication, which is basically they like this particular site, and when they cut, they duplicate that, and then this target site flanks the whole transposon, right? This will be relevant later. But actually, for the talk, I wanted to introduce to you meiotic drivers, right? Which are another type of selfish genetic element, and they characterized by breaking the first law of Mendel of equal segregation. So now Mendel is crying. Because, yeah, basically, if you increase your proportion in the gametes or in the offspring, then of course you're gonna start increasing the population, right? How does this work? Imagine that you have your typical diploid organism, and one of them, well, let's have a cross basically, and one of them is going to be a homozygous for our meiotic driver. So normally, you cross your diploids, you get a heterozygous individual, and in the gonads of that guy, you're going to have meiosis, right? So you have meiosis 1 and then meiosis 2, and normally these products of meiosis should become the gametes, in this case, the sperm. But that driver would somehow inactivate the sperm that doesn't contain it, right? So those are gone. And that's considered meiotic drive by some people. Some people would say that the true meiotic drive should happen during meiosis itself. And that's what some other people would call the female meiotic drive. This is also very classic. You probably know or remember that when you get meiosis in the females, basically only one of the products makes it and becomes the egg, and the rest are discarded as the volar bodies. Volar bodies, there. So basically what the driver does is that it somehow manipulates, say, the centromere or something, so it will be the one that always ends up in the egg. And that's how it produces this segregation distortion. And the one that I just explained to you would be called the male drive, right? Also called sperm killer. Now, a lot of people used to think that these meiotic drive elements were, like these genetic oddities, but at this point, it seems like they are in fact super common. It's more like they are just really hard to detect and study. So basically, you pick a, like a model organism, whatever you like, C. elegans or mouse or whatever, you will find a meiotic driver most of the time. Like, it's very, very common if you look really, really hard. But like I said, it's really hard to find it because, you need to check out the offspring, make sure that there's segregation distortion, that is not other type of incompatibility. But in fungi, this is not a problem because meiotic drive looks a bit different in what we call spore killing. So before we had these diploid organisms, right? But if you are familiar with fungi, you would know that a lot of them spend most of their life cycle in the haploid stage. So when you have two individuals, these now are haploid, they cross the same way as the other one, and now the zygote or heterozygote guy is like super short-lived, and it immediately goes to meiosis. And now the products of meiosis that before were the sperm are now the spores. So what happens is that the driver, now the killer, we call it, somehow kills its siblings. And that is super convenient for us because that means that you only have to go open the fruiting body and boom, there you have it. You can observe it even in nature, which I have. So that's really cool and convenient. This is Podospora again for Linnea, so you get familiar with that. Now, the thing is, not all meiotic drivers are the same. It matters the way meiotic driver happens if we think on the long-term effect in the population, right? So in this little diagram, we have the normal meiosis, where you have well, rather, the pool of gametes is half one allele and half the other, right? In the spore killing case, or even the sperm killing case, you would just deactivate one of the categories of gametes. But if you look at the total number, the actual number of gametes, they didn't change for the driver. They are still the same number of gametes. So that means that it's not actually increasing in population, and we call that the relative drive. And some people have argued that, and I mean, there's theoretical models that suggest that that makes it really weak. It's harder to invade a population because it has less of an advantage, actually, but a lot of potential fitness cost. And on the other extreme, you have female drive, in which case there's, like almost no cost. All of the gametes have the new allele or the driver allele, and that we call the absolute advantage. And that actually is very powerful, right? Like it can, in principle, invade a lot population. So if you have some way to manipulate how many gametes happen, how many gametes occur at the end, maybe the relative drive can somehow shift into an absolute drive. And we call that the killing advantage. For example, say that you kill the spores, but maybe the fungus does something and it makes more spores to compensate. Or there was actually sibling competition. And now if you kill half the siblings, now the siblings that do, or the spores that do germinate, have more resources for themselves, and then they really have an advantage. So in this diagram, yeah?
In the previous slide, so yeah, like for gene drives, like the synthetic type, would that be considered a female drive?
No, it depends on how you make it. There's many designs, right? And I think most of the gene drives are based on CRISPR-Cas and stuff like that. And I think they are, yeah, they are like genes, like it's not necessarily you're gonna put it like somehow, or you have to associate it to the centromere or something. Yeah, I think it would behave more like a relative drive, actually. Yeah, it depends on how you make it, I guess. And also depends on the life cycle of the organism.
Because it's not killing the other gene, right? It's like converting it to the whatever.
I see. You're thinking, yeah, exactly. You're thinking on the homing. Yeah, I guess so. That's true. Yeah, I guess so.
Anyways.
Yeah, okay. Right, so in these graphs, basically they're saying something about the probability of invasion of a killer. In the x-axis, you have the number of generations, and the y-axis, you have the frequency of the killer allele. And the little numbers below are the average probability of invasion based on different efficiency of killing. Meaning, for example, say that you're a very good killer, like you kill all of the other guys that don't have you, so the dark line, then your probability of fixation, say, for example, for the absolute drive, it's really, really high. Or invasion, rather, and fixation, I guess. But if you're a relative drive, then it's really low. And as you become, or you get some killing advantage, this probability increases. Okay, so that's like the general gist of meiotic drive, I guess, and the introduction of spore killing. So now I wanted to tell you what we know about the genetics of spore killers and how they look like in the genome. And for that, I will be talking about Schistosaccharomyces pombi, the fission yeast, Podospora, and Neurospora. I just wanted to make a little stop to emphasize how convenient fungi are for studying these kind of questions. In principle, yeah, basically you have a lot of model species to work with. They're mostly easy to grow in the lab. They have short life cycles, so you can make experiments. They are good for genetics most of the time. And then they have small genomes that have very little T content, which makes it really convenient for genome assembly and stuff like this. So that means that we can get basically telomere to telomere assemblies with not that much effort. And that gives us the power to say many of the things that I'm going to tell you about. Cool. Now, the thing with the spore killers is that if you look hard again, you will find them, right? Like you choose your main fungal systems with one exception, and they will all have meiotic drivers that, like I said, have been published recently. But I want to start with pombi because it's, I think it's easier to understand, or like a good introduction. So pombi, it's a unicellular eukaryote, right? So then basically the cell is just floating there, and then it meets another one, and then they mate, and they form the heterozygote, and that guy immediately goes to meiosis. And so when you look at the ascus, as it's called, this little sac, then you will see that the spores that don't have the killer will like be shrunken and die. And people had done a lot of genetics on this, and basically they discovered that spore killing is caused by a family of genes that are called the Wtf genes. Very appropriate name, if you ask me, but it doesn't mean what you're thinking, because it actually stands for with TF, and TF is a type of transposon, or like the only transposon really in pombi. And the way it works is that you have one gene, but it has two transcripts. And say you have multiple exons, right? But the two transcripts have, or they are different because they have a different starting codon. So one of the transcript is longer, and that one acts as the antidote, and the shorter transcript acts as a poison. So what happens is that when you're making your spores, somehow the poison can, migrate out of the nuclear envelope, contaminate the other spores, kill them, but the spore that actually has the gene still expresses the antidote that cannot leave, and that's how it achieves the spore killing. Now, a really cool thing about the WTFs is that there's tons of them, like tons of them. So if you just pick one strain from pombi out of the collections, you will have between 20 and 40 of them. It's ridiculous. And they are often associated to this transposon, right? So I think it is believed that maybe the part of the proliferation of the gene family has to do with the transposons themselves. But it also has a lot of consequences for reproductive isolation in principle, right? Because if you have 40 of these disgusting things and you just want to mate with another guy that has yet other set of 20 disgusting things, then nothing will survive. And well, to be fair, pombi does a lot of selfing, so maybe it doesn't have to worry much about that, but it is a problem, but perhaps. Now, another curious thing of this system is that it's very easy to create these so-called resistant alleles. Basically, you just mutate the methionine making the poison, and you end up with the transcript that it's only the antidote. And then basically that one now becomes selected for because it removes the segregation distortion from the homologue. And that one goes to fixation now. So in a sense, the host can recruit these resistant alleles to fight back. Yeah. And another funny thing is that you can also have gene conversion between the copies, meaning that, and this has been seen, that they can shuffle exons, and then suddenly one WTF will be able to suppress the action of the other WTF while still being a driver itself. So yeah, that gets complicated. So that's the WTFs. Very, very funny. But now we're going to Podospora, which is my guy. Yeah. Podospora and Fungi are, or Schizochromyces fungi, are extremely far away in the phylogeny. If it's extremely far away, 500 million years divergence, right? They have nothing to do with each other, basically. And yet, the spore killer in Podospora work in a very, very similar way. They have evolved, they are not homologous. They're completely fungus that done herbivores. It's always easy to equidate, but as far as we can tell, there's no like different transcripts happening. It seems like the same protein somehow is doing both the jobs of being poison and antidote. And we know that because our collaborators have done, you know, like point mutations. They have, for example, they have that you can break the killer part, and then now you create a resistant allele, just like you had in the WTF case. Or you can kill the resistant allele, and now the gene is suicidal because it kills itself. And in the case of Podospora, we know that there's at least 4 homologs segregating around. One of them, SPOC1, is actually in another species, but SPOC2, 3, and 4 are in the same species, in the same population. And that's something different from the WTF case, because you know, with this yeast stuff, you have like one strain here and one strain in another continent, so you don't really know what the population dynamic is. But with the SPOC genes, we have no idea what happens in all the other places, but in this one population in the Netherlands, we know a lot. And for sure, these killer genes are all segregating there. And what's funny about them is that they all can kill each other. So if you have SPOC2 and you mate with another guy that has SPOC4, you would kill each other. And funny enough, SPOC1, which is in another species, can kill everybody. That's like the king of everybody, except maybe SPOC4 because they are mutually resistant. But again, a given strain, so you just go into the field, you sample one poop, and you find one Podospora, and it can have, you know, 0, 1, 2, or 3 SPOC genes. And so that creates a bit like a hierarchy of how good of a killer you are in the population. And this, like I said, this population has been studying for a while because if you want to know the story, basically there's this professor in Wageningen in the Netherlands that has a genetics course, and every semester he used to take his students to go sample poop to find Podospora strains. So he has been sampling since the early 90s, which is super cool. And thanks to this data, I mean, we sequence it and a lot of things happen, but we know now that SPOC2 is almost fixed, like very high frequency in the population all this time. Like, and actually I sequenced, well, I sampled up to 2017 as well, and it's still the same picture. While SPOC3 and 4 are in the like lower, almost intermediate levels. Now, we start, yes?
What happened with SPOC1 there?
SPOC1 is another species, right?
Oh, right. Okay, sorry. I'm with you.
Yeah, no, that's also, I would like to know what happened to SPOC1. So basically there's only 3 strains from the other species ever sequenced. They all have SPOC1, so maybe SPOC1 is fixed, but there's not much more. Basically, there was only one strain for that species for the longest time, and then I stumbled into some more in Wageningen, and I sequenced them because I thought they were Anserina, and they weren't. But yeah, so maybe it is fixed. But unlike SPOC2 and maybe SPOC1, there's some SPOCs like SPOC2 and 3, sorry, 3 and 4, those are different. They do something different from the WTFs as well. Basically, when you, again, sample one strain from the wild, you would find, say, Spock 3 and 4, and maybe it's in chromosome 3. And then you sample another strain, and now those two are in chromosome 5. And now you sample another one, and now it's in chromosome 1. And you're like, how? What's happening? Like this is the same population, right? How is that happening? Well, it turns out that the Spocks are not alone. They are inside this much larger haplotype that we call the Spock block. And actually what's moving is the whole block. That whole chunk is moved in different places. Now, this is really strange because the Podosporas have extremely low genetic diversity. It's like the same things. They are really so similar, it's crazy. They are extremely synthetic as well. And their genome is really small, it's 36 megabases or so. But the block can be up to 240 kb. That's really big. Like if you look at the proportion of the genome, it's kind of big. So what is that thing? Like if you want some perspective, think that normal genes or even transposable elements, they are like no more than 15 kb. So what the hell is that thing, right? Well, long story short, it is a transposon. It's just a really, really big one. And we know that because it has target site duplications, as maybe you remember I said at the beginning. And we believe this transposon is mobilized by a particular gene that we call KIRK. And KIRK is a tyrosine recombinase, so that's probably what's doing the cutting. And the whole element we call the enterprise. And the enterprise, occasionally, at least in Podospora, seem to have captured Spock genes. So there's KIRK, Spock, they travel in the enterprise. That means that the block have this dual dimension to selfishness, right? Because it can do meiotic drive, but it can also do transposition. And that's like, wow. Well, it's like, like we call it hyperparasiting. And going back to this whole situation about absolute drive and relative drive, we were really interested in seeing if there was some fitness effects, right? So we designed a little experiment. You don't have to worry about the complicated names, but basically what we did is that, remember I told you Spock 2 is basically fixed in the population, right? So we took one guy that is typical, it has Spock 2, and then we introgress the block into that background, that genetic background, and one in chromosome 3, and then we introgress it again, but now in chromosome 5, sorry. So they should be the same except for the block, right? Basically what we did is that we crossed them to each other, and because Podospora can self-fertilize, we also just let it do so. And this is what we got. We tried to measure other fitness traits and we found nothing, but this—
Wait, Lore?
Yeah?
I see still Podospora and Asteria, the slide. Are you going on to other slides or is that on my end?
Wait, so you don't see anything now?
I'm fine, I see everything.
Oh, okay, that's me then.
Yeah, we also see it.
Okay.
Okay, wait, guys, what are you seeing? Now I don't know what
I see a horse on the top right.
Oh my God, I've been far from that for a while.
We're
Right.
seeing chromosomes. We see
Yeah.
No, we are good. We are following along with you. The slides are moving as expected.
Okay, so it's just me. All right.
Oh,
let's
no.
see. I will stop this recording.
So the tyrosine recombinase has it's a particular type of protein that has been found in many other organisms, including bacteria, but it's associated with a specific type of transposon called krypton. And the kryptons are not very well studied either. So actually KIRK stands for krypton-related, no, how is it? Healing-related krypton, there you go. So that's how we also figure, okay, this really looks like a transposon. Because this particular type of domain is associated to these particular types of very unknown Ts. But exactly what it does, we actually don't know because it's not, it's not trivial maybe to do the functional analysis for this. because we don't really know how it works. We think it might be cut and paste, but we don't know.
Okay, cool.
Yeah, so yeah, but that's for sure something that it would be nice to characterize. I'm sure Aaron is going to be super into that.
I have a quick question about like your methods. So you say
Yeah.
that these things are really, really long. How do you figure out what chromosome they've jumped into? Do you sequence?
So remember I told you that they are the fungi are completely synthetic, right? Like you can take chromosome 5 of one strain with chromosome 5 of the other strain, and then you just see this block there that has no homology with the other one.
But you
so
sequence.
Really, yeah, we like I said, we have telomere to telomere assemblies, right?
Right. Okay. So then you just do like parallel sequencing, and then you map to the.
Yeah. Well, so we have how many? I guess like 9 very high quality strain sequence. You know, like full chromosomes.
Yeah.
We just have to make a genome alignments, basically.
Okay. So you just like do massive parallel sequencing, and then you hope to cover the junctions.
I mean, the junctions, what do you mean? It's just like, it's really, there's no problem with the sequencing. It's just like you have plenty of reads going across.
Yeah, I guess I just mean that like a lot of the reads are going to be outside of the transposon. A lot of them are going to be inside the transposon.
Yeah, but the strain is haploid, right? So I just need to, like, it will have the transposon or it won't. And then when I'm sequencing, it's just like, it's not like I'm pulling them.
Okay,
It's
okay.
individually sequenced.
Good.
So it's very straightforward in that sense. Okay. So should I continue though? Okay. Right. So like I told you, we had this little experiment where we had the base typical strain and then we introgress the block, right? Okay. So in this plot, what you see is that on the x-axis you have different combinations of mothers and paternal contribution. And then on the y you have the number of spores produced in total. And first, let's look at the blue comparisons. Those are basically self-fertilization. So there's no spore killing, actually, right? It's like everybody has a killer, so there's no killing. But still, if you have no block, you have more spores than if you do have the block, at least in chromosome 1. So that means that just carrying the block alone, without killing, that's deleterious. Something is wrong. That's not significant for chromosome 5. So actually, maybe it matters also where the spore block is inserting. So maybe for whatever reason, it's worse to have it in chromosome 1 than have it in chromosome 5. That's the first conclusion. The other conclusion is that if you look in here, for example, you can see my mouse, right? I'm crossing the block in chromosome 3, Jesus. Chromosome 3 against no block, but the mother changes. So the first strain here is the mother. So at the beginning, say the mother has the block or the mother doesn't have the block. And that makes a difference. So that means that there's a maternal effect also of having the block. People look confused. You're muted, Lina.
Sorry. Can you just tell me with each of these points in your plot?
Yeah, it's a cross.
Is an independent cross, but you didn't question. Did you the genetic transformation that you do where you insert the spot block?
It's not a transformation. It's a back crossing.
Oh, it's back crossing.
Mm.
But what I mean is, did you do the back crossing like in 20 different like independent replicates, or did you do one back crossing and then you took that one line and crossed it with everyone else? I understand also that in this case it doesn't really matter because you basically don't have any genetic variation in your genomes as far as I can tell. Mm-hmm. So each point is one block. And how do you, when you say yield,
mm-hmm. How does that look like?
Yeah, how do you like, is that
so basically, yeah, so basically you have a plate, right? Your little petri dish, and you put your maternal strain, and then you fertilize it with the what we call the sperm equivalent of whatever strain that you want. So it could be itself, like its own sperm, and that's self-fertilization, or it could be somebody else's. And then you just let it sit, and fruiting bodies will develop, and then they will shoot the spores into the lid because they are phototropic. So they like to shoot into the light. And then you just take the lid and you recover the spores, and then you do some very nasty chemistry to try to remove them because they are sticky, and then you count them.
So, and when you say maternal and paternal, do you refer to the mating type, or do you refer
No.
to actually who is the receiver?
Who is the receiver? Because there's fungi are hermaphroditic, right? So the mating type is unrelated, decoupled from female or male function.
Yeah, no, I just meant in the back crossing.
So the mating type doesn't matter because it's not linked to the block or anything. Okay, so maybe to answer your question, in this case, we had all of the females are going to be one mating type.
Okay, yeah, exactly. That's what I was getting to. Okay.
Yes, yes. We didn't do it the other way. That would have been great, but it was just too many because we were dealing at that point with like 1,000 plates or something.
Yeah, that makes sense. Yeah.
Yeah. No, but that's unfortunate, actually. It would be great to test it. But we are assuming that there's no effect of the mating type. And there's reasons to believe that that's the case. Okay. People are good? I'm going to assume that yes, given the silence.
Perhaps a stupid question, but what is
No, I'm sure it's good.
meaning? Is it like the S is like, because
Yeah, the names are confusing. That's why I didn't want you to care too much about it, unfortunately.
Okay.
But so S is the name of the strain. It's just a name. It doesn't mean anything in particular because there's like A, B, C, D, you know, like so many strains. And S happens to be the reference genome.
Okay, all good.
So that's why it's called S. And it also happens to be the typical strain that only has SPOC2 but no block. So that's why we use it, but it doesn't mean anything more than that. But it's not a stupid question. It's actually a really, I think I spent like 1 month in my PhD very confused about it as well. Like, who is that? Okay. Right. So the conclusion was that the block is deleterious. There's a bit of a maternal effect, and something that is hard to see here, you will have to know a bit more, but one very important finding from this experiment was that when you do have spore killing in these red crosses, the amount of spores that you recover are more than what you would expect given the killing efficiency of the spore killers. And how do we know the killing efficiency? I'm not going to explain that because it's really complicated, but it has to do with the recombination rate and the genetics of Podospora. That is really out of the scope of this talk. But basically, we know, okay, if the block is sitting in this location in the genome, it will kill 50% of the meiosis. We know that. And then what we see is that there are more spores than that. So that means that somehow the fungus is compensating for the loss of spores, meaning that, in fact, there is some absolute advantage. Boom. That was very important for us to know because there's basically no evidence of this at all for any spore killer. Yeah, so we have some absolute drive, yo. That's very important. Now that takes us to another discussion that this is not solved at all. It's just more like philosophically, you can ask, are they working together, the enterprise that is Kirk and Spock, or are they working against each other, or one of them is parasitizing the other, right? In other words, you could think, oh, maybe for the killer it's really good because you know, you're driving, but also you're creating more copies of yourself, so maybe that's convenient. But you can also think the other way, because the Spock genes that are not in the block are near fixation, right? As we just discussed. So maybe just being your little Spock gene that's actually not deleterious, and you just go to fixation really fast because you have no trouble. But now that you're sitting in the Spock block, now you have deleterious effects, and now you cannot drive as much. But for the point of view of the transposon, it's really good because, you know, I'm so fat, maybe I cannot copy myself so effectively, but hey, I have meiotic drive now. So that's something that we don't know, but I think is an interesting aspect of the system. And actually, while all of this is happening, it's not like the host is doing nothing, you know? So many, well, eukaryotes in general have some form of genome defense mechanisms. But in fungi, we have one called, called RIP. RIP stands for repeat induced point mutation. And what this mechanism does is that basically whenever you have 2 repeated sequences, whatever they are in the genome, RIP will find them and they will, well, RIP will mutate them to death. Basically, it finds all the, well, not all of them, but often it likes to mutate all C's to T's. And then by chance, you will just break any coding part there by just mutating it a lot. And that means that all the repeated elements in the genome are often very low on GC content. And so whenever you, I mean, if you go into the genome, you may find the, you know, the corpse of an old Fock block, and that one often has very low GC content. And we can recognize it because it has some, you know, cadaver of some genes that we know are in the Spock block, including the Spock genes. At the same time, there might be a bit of a disadvantage of having RIP. And that's because if you can mutate anything that is repeated, that also applies for genes, right? And if you, I don't know, if you read Lynch or something like this, you would be like, oh yeah, you need gene duplication, or that's very important for making new functions and sub-functions and so on. But if you repeat your genes here, you kill both. So actually in this type of genomes, it's not so common to find these gene family expansions as in other groups. So that's inconvenient, perhaps. And something that could also backfire is that maybe you mutate your Spocks that are repeated, but maybe by chance, those mutations actually create new killers, you know, because you have one particular killer, but then now that you mutated it, now maybe this one is no longer reacting to this version of the gene, and now it can drive on its own. So that could also backfire in that way. You can also go other ways, right? You could be like, oh, what if the enterprise actually has other type of cargo and it could be advantageous? And there's a little bit of evidence of that. There's another big fat transposon called Hephaestus. I cannot English. Hephaestus. And that one has been shown to have some genes that are good, I believe, for heavy metal metabolism or something. And then Aaron went and checked, of course, and one of the genes at the very beginning is a Kirk. So basically it looks a bit like Hephaestus may be a type of enterprise. So, you know, it seems like you could go both ways, negative and positive. Okay, I don't know how long have I been talking, maybe a lot. I just wanted to maybe drop really fast the case of Neurospora. If you ever, maybe you remember Neurospora from your classes, like it's the original eukaryotic system, you know, that's how, well, it's a model organism that is very important. And it has other type of meiotic drivers called SK2 and SK3. But unlike the WTFs and the Spocks, these ones are really big haplotypes. And this type of architecture is actually super common for plant and animal meiotic drivers. We can discuss why that is. And in this case, you have 2 genes that are doing the functions, right? One is the killer and one is the resistant or antidote gene. And because you need to have them together, otherwise you create, you know, like suicidal alleles, you need to somehow suppress recombination. Now my fridge is sounding, I hope you don't hear it. Anyway, in the case of Neurospora, what happened is that there was a huge blockage of recombination that covers half the chromosome. Maybe you've never seen this kind of plots, but basically there's like, this is the chromosome 3, and it's an alignment of a killer version versus a non-killer version. The green lines basically mean that it's aligned and in the same sense. The red lines are aligned but inverted in one of them. And if you have no lines, it's because there's no homology in between. So something is there that is not in the other. And in this case, we know what the resistant gene is, what the killer gene is, and they are both surrounding the centromere. And that's SK2. And there's also SK3 that is in the same place. although it seems to be using the same resistant gene, is using another unknown killer gene. And the inversions are totally different. So actually, it seems that these two evolve independently using very similar mechanisms. And yeah, they're rather massive. So that's also funny. And when you look at the GC content around them, you see all these drops in GC, and that's because it got full of transposable elements that maybe cannot be purged precisely because there's no recombination, right? There's some evidence that they are basically degrading. All right, so that was the main 4 killers. And basically you have like these typical architectures, perhaps. One would be your Spock genes and other types of genes that, maybe they're just hanging there, maybe a bit associated to transposable elements, but in principle, they don't need them. The Wtf would be an extreme case where you have a lot of them and they like to be with the transposons. Then you have SK2 and SK3, where the transposons are just coming, independently because the place is so large and there's no recombination. And then there's the super strange case of SPOC3 and SPOC4 being now part of the transposable element. One could speculate that maybe the little guys have less deleterious effects, maybe being small and single gene allows you to just go to fixation really fast. Maybe being large makes you more deleterious because you're more prone to accumulation of crap. Could be. And yeah, so maybe I can just summarize. Basically, all of these 4 killers have evolved independently, right? So that suggests, because they are very in different positions in the phylogeny, they're very apart, right? So that suggests that basically spore killers could be everywhere. And you could also extrapolate that to other eukaryotic groups. There's no reason why it wouldn't be like that. And from what we learn, it seems that they are affecting the fitness of the host, they are affecting their genome composition and architecture, and that often transposons are involved. Yeah. So I think that's all I had to say. Yeah. That's it.
Cool. Does anyone have any questions? I think Augustine had a couple questions in the chat that you might not have seen.
No, I don't see anything for the chat, but maybe hope you can read them to me because I cannot see anything.
There was one like before I cut out, so I can't see that one either. But there's a second one that said, what are the proposed mechanisms for the maternal effect?
Oh, we have none, absolutely none. Yeah, I don't know. I mean, it could be, for example, that the Spocks. So one thing that we saw is that the Spock seems to be expressed vegetatively as well. So maybe they are more like a cytoplasmatic thing staying there. But that's the Spock, not the block. So it's unclear. Another observation is that the Spock block that is deleterious or significant at least is sitting right next to the ribosomal operand, like the tandem repeat operand. So would be interfering with some heterochromatic structure or something like that. But basically, we don't know. That's a good question.
Thank you.
So what was the other question?
The other question was, have the WTF genes been co-opted from another gene, and what would be its original function?
Yeah, that's a also excellent question. It is not known because, unfortunately, Fungi seems to be sitting a little bit alone in the phylogeny. There's nothing close to study, so we actually don't know. But something really peculiar about the function of these genes is that it seems to be very universal in the sense that they have inserted the same WTFs in the other yeast, the Cerevisiae, which is super far away, although they are both yeast. It still works. And during a talk here in Uppsala, the main PI said that they think they also put it in animal cells and it also worked. So it seems that it seems like the WTFs are making some aggregates within the cell, and those aggregates, I suspect they just are garbage blocking stuff, and that becomes toxic somehow. That seems actually universal enough. Yeah. Hope that answered your question.
Yeah,
No,
thank
no,
you.
it didn't actually. But another thing I should say is that for the spokes, they also seem to be really widespread in the phylogeny. But we have absolutely no clue if they were in the past part of the normal cells or normal function, or if they had been domesticated. We have no evidence of that. No. But also, it's, how to find it is going to be hard. Yeah. Oh, no more questions.
I have a question.
Also, yes, please.
How does the RIP mechanism work? Like how does the genome say like, oh, there's a duplicated gene? Or
Yeah, that's also rather fascinating because I myself don't really understand, but people do. It seems like, it's gonna sound weird, but it seems like it works like BLAST. It's sort of
exactly, yeah, but it's yeah.
Yeah, but molecularly. So what people have done experiments about just how much homology you need for RIP machinery to find it. And yeah, you seem to need certain number, like a k-mer of size. And those k-mers have to be at a certain distance from each other for the homology to still work. But I don't know, maybe I don't know, but it seems unclear to me at least how exactly the machinery can tell that, this piece of DNA in this chromosome is repeated compared to that other chromosome far away,
Yeah, it seems like it requires some memory, or I don't know.
I don't know. Of course, you can imagine that there's some interaction, three-dimensional interaction between the chromosomes, and maybe that's how, but I don't know. Yeah, but I think that's also super crazy.
Yeah, thanks. It was really, really cool talk.
All right. Are there any more questions? Sergio.
Can I have a quick question?
No, next question.
Yeah, based on the theory. No, I was just wondering, in the spore plug, since it's very long and it has like many motifs and so on, I was wondering, if you look along the genome, you can find the, I don't know, maybe an ensemble fragments of the spore plug?
Yeah. So maybe I can stop sharing and look for the figure. Wait. Wait. Just that the paper is still not published, and then I forgot completely where I left everything. Oh, wait. So well, just to answer your question, basically, if you check the alignment of the block against the whole genome, you will see links of alignments of everywhere. That's partially because it also has a lot of TEs that are just in the genome, but there seem to be some homologs in the host genome. So that could go both ways, right? It could be that the genes were moving to the Spock block, or that the Spock block brought them and then they move into the host genome. Could be both, and they sometimes look like very, very similar, as if they were new, or they look very diverged, but there's also RIP that breaks everything, so it's hard to say.
But then in terms of the GC content that you mentioned, that you expect a reduction or increase of GC content, do you see that relative to the homologous sequences, those homologous sequences?
Yeah, it's hard to say because, so if it's a TE, you cannot, there's no way because there's other copies in the genome, so of course it's going to be ripped. And if the genes are diverged enough, then RIP stops working. So I think there's a threshold, I don't remember, say like 70% identity, and then RIP it no longer thinks it's the same thing. So it's not obvious, at least.
And just one last question. In the sequence, you have whole genome data, like I guess that you reconstruct the whole haplotype and so on those samples. Can you maybe use this new information in the block to infer maybe ancestry?
No.
Like how old was the insertion between them and so on?
Yeah, we tried, but that's not. So if you align the blocks to each other, they are very, very similar in sequence, but they are rearranged. And it was not obvious. Yeah, like we tried to be like, oh, maybe this is the original copy and that one, but it wasn't easy to reconstruct, and in the end we gave up. So there was no sequence divergence as much as content and inversions. Yeah, it wasn't, we tried, but we couldn't.
Not even within the repetitive sequences, if you extract the little t's inside and outside the genome to try to see if you have certain sequences, like to say at least the sequence going inside several times or the way around.
Yeah, it didn't pop up as something obvious. It's more like if you have repeated thing, it would probably be a transposon. And then you have other 30 copies, and then you're like,
yeah, but I guess with long reads you should be able to call variants in those repeats, right?
but what do you mean by variants? Because you're just like, you can just look at the same genome, right?
Well, let's say you have a transposable element, you extract all the sequences of that transposable elements and then you align them, every single copy of a particular family of transposable element.
Yeah, but so we have done that for other reasons, and there was no obvious pattern. Having said that, the beginning of the enterprise itself actually gave some signal. There seemed to be like 2 types. That's not that we didn't publish that anywhere, but there were like 2 types, and so maybe you could see some signal there being like, this group is the one that seems to be moving this way, the other not so much, or maybe the other one is just broken. But the T's themselves, it's too messy, in fact, because we have too many types, right? It's not Fungi. and the phylogeny, because RIP breaks everything, and then the T's are nested within each other, is not easy to recover an alignment. Sometimes you can, but it's not very easy. Oh my God, I just noticed that Arash is here. Wow.
Special guest.
Yeah, I mean,
are you planning to continue working on this stuff?
No, not at all. I'm out. No, Linnea will take over. No. Actually, this whole genome conflict situation was half of my PhD. The other half, it's more on speciation and allorecognition and other topics. And so I'm gonna just focus on that, try to finish that. But I still don't have a job, by the way. Looking for postdoc.
Giannis is here.
So, okay, yeah, hi, Giannis. No, so basically, I don't know what's gonna happen in the future. But when it comes to Podospora, at least I am gonna be working on speciation and other topics. And another PhD student in the group now started working with the centromeres of Podospora. So that's another path.
Are you going to keep working on fungus?
Not necessarily, no. I could do whatever. Yeah.
Cool. All right. Anyone have any last questions, comments?
I have a few pages of questions.
Oh God.
But my connection it's the lowest quality connection I ever had for a while. So probably, I don't know. Can I send you my questions by email, Lorena?
Yes, of course. Sure.
Okay. I mean, I can try one at least. Can you hear me? Okay.
Yes.
Yes.
Yeah.
Okay. So is the association between meiotic drive and transposable elements general or restricted to fungi?
I don't think it's restricted to fungi. I don't see why would that be. But I think we have better genomes than most other people. And so we can see it more clearly. Probably if you would work with other meiotic drivers in, say, plants and animals, probably. You could see these, but maybe not
Well,
as in your face. Say again?
There are meiotic drivers in Drosophila and mice, but
Plenty, yeah.
I don't know if they are associated with transposable elements. It would be easy to see.
Yeah, in that case, it's a bit, there's a complication, which is that meiotic drivers in Drosophila are often associated to the sex chromosomes. So, of course, there's going to be these there. But then people think that they are in the sex chromosomes because, you know, lack of recombination and because most people working on meiotic drive are used to thinking in the two loci systems where you require suppression of recombination. So yeah, I don't think TEs are like a thing as much.
But okay, but so the other side of the question is why do you think TEs are so often associated with drivers, at least in the systems that you showed?
If I just speculate, my gut feeling is simply that if they hook to each other, then the drivers can just take advantage of the diversification. Basically, if the T can make another copy and that copy diverge functionally, then boom, you have another driver, right? So maybe whenever that happens by accident, it's successful. So that would be my gut feeling. Yeah.
Okay.
Why it happens.
Okay, thank you.
Well, thank you.
Alrighty. So I guess we'll finish up here. Just another reminder, if anyone wants to present next week, please let me know. Otherwise, I guess we'll probably be taking a break. Ari, are you free next week? Like at this time?
I gotta check my schedule, but if there's something up, I can block it so that, you know, nothing.
Okay, because like the presentation you gave for our lab meeting was very well received, so you could just do the same thing for memes.
Sure, be happy to.
Okay.
So Wednesday
at like 1 p.m. EST.
My schedule is clear.
All right, awesome. I'll organize stuff with you later.
Awesome.
Cool. All right, we'll be meeting next week. Thanks, All right, cool. Thanks, everyone.